What I'm going to talk about is something that's catching everybody's fancy these days, which is the hope and the hype of stem cells. The cell, as you notice, is the fundamental unit of life. You can't do without it. If people define life, you need to have two components and two definite, irrevocable conditions for definition of life. You call something living if it has two properties. One of it is it must make more of itself, self-renewal, reproduction. And the second, in order to do so, it has to react, interact, transact energy, material between itself and the environment. One won't do, both must be there. A virus has information to make more of itself. It can't transact. It has to be a parasite on somebody else. A red blood cell that most of us have doesn't have information to make more of itself. It can transact business with. So that what you need in a cell is therefore two things. Make more of itself, and to do so, it must metabolize. And cells go ahead, assemble to make tissues, and tissues make organs, and organs make all of us, right? Even that is a living form, single cell. Many of us are not that. Occasionally, for example, each cell may have two cells put together, and that also is a living form. But you notice the information content in there is what we define as the number of genes. In the genetic information, you talk about 0 and 1 in terms of information content as a binary. We talk in biology of information in terms of quaternary. You call bits, I'll call it quits. The information is put in there, for example, 0, 1, 1, and so on. In our case, it turns out to be A, G, C, T, and so on. You put on a sequence of A, G, C, T, whatever, and then you read it out, you get information out of it. That's what you call, that sequence is what is referred to as genes. You notice even in that little thing, which is the E. coli bacterium, the number of genes is pretty large. Or the order of about 15 to 18,000 is not too bad a number. Most of us have, the number of genes that we have is approximately about 30,000. The number of genes that a plant has is probably more like about 35,000. The number of genes that the spider has, a little more than what we have. So it is not what you have in terms of information content that's important. It's how you're going to handle it that's important. Look at this. This is the darling of a variety of cell biologists. This is from there to there no more than about a millimeter or so, 2 millimeters long. And it packs in itself, you notice, something like about 1,000 cells. We talked about E. coli as one cell organism. We have a 1,000-cell organism here. The number of genes in this worm is 23,000. And the question is, if you call it a living form, how do you think of it to make more of itself? How do you make babies? How do you reproduce? And by and large, many of these happens to be by sexual reproduction. So you need to have a man and a woman. You need to have a male and a female. Occasionally, you can also have a male and a female. It's not very often that it happens in life forms. Occasionally, you may also have 3 parents to make a baby. We don't do that, but anyway. I hope we don't do that. And what you do is, by sexual reproduction, notice, regardless of whatever you do, this is exactly what's the kind of a thing that's going to happen. And here is what we do. The egg of the female, you can see, is on top of the ovary. The sperm meets the egg. The sperm contains the genes of the male. The egg contains the genes of the female. They must fuse together, fertilize, and you make a baby. The baby, essentially, you notice how it is. The sperm penetrates the egg. That's the moment of conception. Penetration actually has happened. And once it's done, we fertilize. And that divides. One becomes 2, becomes 4, becomes 8, and so on, and this is what is referred to the endometrium. This is the one that must go and attach itself to the appropriate implantable thing and so on, and that's how the baby is born. It's that single fertilized egg that gives each one of us 210 different types of cells that make the body. Blood alone has 6 different types of cells. All this has to come from a single cell that is fertilized. That I think is the essential part. Notice the number of genes that we have is about 30,000, but the number of proteins we have is far more than that. Genes are information strips. Proteins are action molecules. Notice the number of action molecules is about 3-fold higher than the number of informational molecules that you have. So you need to have duplication, triplication, and so on that should happen in here. But the idea behind all this is we are all born out of a single fertilized egg. And that fertilized egg, notice, within a little term of the order of about 8 weeks or so, might already produce all the organs that you have in there. What happens? Let's go in here. When the embryonic cell, that's a fertilized cell that became 2, 4, 8, and so on, we'll call that the embryo. Once it goes on into approximately about, let's say about 1,000 cells and so on, you can start calling it an embryo. It can do everything. It can produce all the 210 cells of the body. But you notice you had to ask that question. If I have a white blood cell which is different from my muscle cell and that's different from my nerve cell and so on, they all have exactly the same information. The DNA in there is exactly the same. And yet the information which is contained in there is processed differently. Some information is stopped. Some genes are silenced, some genes are activated, and so on. What's the mechanism? This is by and large the question that is addressed by a large number of cell biologists today. And that's a question that was asked by John Gurdon, a graduate student in Cambridge in the 1950s. He asked this question. I take a tadpole, it becomes a frog, you go on through the cycle over and over again, and the information there is identical. The genes are the same, and yet, do they all have the same information, or genes lost or silenced as you go along in development? So what he did was, he removed the nuclear material which contains all the genetic information inside a cell from the late stage embryo of one frog, took the egg cell of another frog, removed the nucleus from there, put this in there. In other words, it's a donation of the genes of egg A into the genes of egg B. Out comes a tadpole. Nobody would believe him at that time. And he said, all right, if that is so, why do I take just an egg cell? Why can't I take any cell? And he took the skin cell of a frog, used exactly the same kind of a thing. From the skin cell, he removed all the DNA, put it in the enucleated egg of another frog, and came out with the tadpole and frog. What does it mean? The genome, which means the entire genetic content, is the same in all cells, but when cells differentiate to produce different types and so on, some genes are silenced and some are activated. The embryonic cell is totipotent. The differentiated cells are not totipotent. This gentleman, Sir Martin Evans in Cardiff, said if you can do that with frogs, why can't I do that with mouse and rats and so on? And he actually did that. Martin Evans isolated the mouse embryonic cells, and John Gurdon isolated the tadpole cells. Another man called John Thomson isolated the human cells of the same kind. So we are talking about embryonic stem cells. How do you get that? You get that from here. You can take any one look at that cell over there, the fertilized egg, which now has divided into, let's say, 8, 16, 32, go on up to 108 or so. You can pull out one cell from there. There is no deficit on the other 107. They make one more, but they become completely 108. You got yourself one embryonic stem cell now, out of which you can make another baby. This is exactly what happens in vitro fertilization clinics all over Hyderabad. The second meaning of that is we can therefore inject nuclear material from one cell into the emptied egg of another cell and create a whole animal. This is no longer male cells and female cells, no longer sperm cells and egg cells. Remember, this is really somatic cells, one that I can pick up from my skin or whatever. And that is therefore referred to as a somatic cell nuclear transfer. Question is, if I can do that with a tadpole, I can do that with a mouse, can I do it with a sheep? That was the question that bothered this gentleman called Ian Wilmot and Keith Campbell. What they did, this experiment here, and this will set the stage for what we are going to talk about. You inject the nucleus from an adult cell that you remove, put it on into the egg of another cell, you have removed the nucleus from there. So you are actually doing a somatic cell nuclear transfer. Allow it to develop. You made yourself a fertilized egg now. Take that fertilized egg, put it on a surrogate mother. You can now create an entire baby. Let's put it this way. I'm going to take, let us say, an egg from that one, from the fin dorset. Egg cell is removed. I'm going to take from here, which is the nucleus donor, from the udder cell of this one. Take the DNA from there, put it over here. It goes through, cell fusion has happened, it's ready. Go to a third mother, would-be mother, surrogate mother, insert that embryo on into her womb. Exactly what happens in vitro fertilization. Out comes a baby. That's Dolly. The mother who gave her entire genome, which came to her from her father and her mother, right? This is the donor mother. This is the recipient mother. This is the actual generating mother. Who's the mother? Who's the father? Who are the grandparents? That lady or that sheep that actually gave her entire genome, gave it because she got it from her father. So Dolly might not be the daughter of Molly. She might actually be the granddaughter. So you got a problem in here, you see. That was possible because embryonic stem cell generated all the cell types there. Okay? We can do one other thing. More recently, it has become possible for us not only to use just the embryonic stem cells, but take, for example, cells from the muscle or from the eye, places like that. They also are able to generate. They won't make a baby, but they can actually go on to make other kinds of cells and so on. They are not totipotent. They are only pluripotent. Some cells are only unipotent. They'll only make more of themselves. But some cells, depending on what medium you put them in, can make other kinds of cells and so on. This is the kind that you're interested in, because suppose Smita talked about how Kannappa Nayanar had to go and remove his eye and put it on the Sivalingam. You don't have to do that. We'll show you how. I won't spend too much of time on this one. So can I take one such cell and actually start, depending on how I'm going to incubate it in what medium, Can I, for example, make bone cells, nerve cells, heart cells, and so on? And the answer to that is yes. So just like a plant has a stem out of which everything else is made, this cell is a stem out of which many other differentiable cells are made. Particularly, it's possible for us to no longer go to the embryo, because Bush didn't want you to go to the embryo. You can actually take it from, for example, the bone marrow. The bone that you have is not solid, it's actually a tube. Inside the tube, there's some remarkably good material which contains a lot of stem cells. One kind that goes to make blood. The other kind that goes to make muscles and so on. So these two parent lines, one is called the hematopoietic, which means that will make all blood. The other is called mesenchymal, that means it can make all other kinds of tissues. You can actually go, many doctors today do it. They take a little bit out and go ahead and do the treatment. For example, Professor Maman Chandy has been doing it for 30 years. You go to a clinic because you had radiation therapy for cancer and so on. In that process, you also lose a lot of hemoglobin. You become severely anemic. What you can do is go on to the same patient's bone marrow, take these cells, go to the laboratory, make more and more of them, inject it back to the patient. That is one kind of a stem cell treatment. We had known it all these 30 years, but we didn't use the word stem cell at that time. They're simply called bone marrow. Is that what it means? It's also possible not to take the blood-making cells from the bone marrow, but also take other kinds, and that's what Dr. Venugopal did in All India Institute. On a patient who was on the operation table because he had suffered because of myocardial infarct, that means the heart muscles have split a little. Can I take the bone marrow, separate not the blood-making cells, but the tissue-making cells, the mesenchymal cells, and put them on right where the infarct has happened? And the answer is yes, you can. And the patient recovers. 41 patients. Long term, 4 years later, there has been no difficulty. So it's possible, therefore, to use bone marrow-derived cells as stem cells. We'll now come to Kannappa Nayanar. Two of my colleagues at the Institute, Geeta Vemuganti, she's a biologist. This is the Dr. Sangwan. Here is the eye, you notice. That part is the cornea. Occasionally it gets damaged because of fire, acid burn, things of that kind. You can't put in a corneal graft because the wound is so deep. But then we discovered that it is possible just around the cornea, where the white of the eye meets the transparent part of the eye, that's called the limbus. That stores stem cells. You can actually go and harvest a bit of that, take it from the patient, biopsy, go to the lab, make sheets of the corneal epithelium, differentiable because it's a stem cell. You see, from the good part of the eye, he actually removes a little bit of the tissue from there. There is no deficit because it grows back. Just like when I shaved this morning, I had a little cut. But in about an hour or so, it grew back. The cornea is no different from a skin. So he gave that little part from the second floor. We brought it to the fifth floor, and we cultured it on a little plate like this. Actually, the plate here is actually a skin. It happens to be the amniotic membrane or the placenta membrane, which is very, very good. And you can grow it to produce something of this kind. In about 8 days, those little cells give you a nice, transparent, stitchable, functional sheet. Not the whole cornea, the sheet. And now you can go on, this is how actually it is cut by our students, and they do the entire thing. And we produce what the properties is and so on. We won't worry about that. This is the bad eye. And what Dr. Sangwan does is now remove all the scar tissue. There should have been no scar. Because there was a burn, there was a scar, just like your Sivalinga. And there should have been no blood, because remember, the front of the eye has no blood. But because there has been a scar tissue, there was blood in here, and he clears all of it. In the meanwhile, we have already made the sheet, and we'll bring it in a nitrocellulose membrane paper, give it to him, and after he cleans the Pannus tissue or the scar tissue, he's going to put all over there. I'm taking an extra minute, I hope you don't mind. Now, that's the one that we made in the lab. You remove and you stitch it just like you stitch a regular cornea. And cover it with a bandaged contact lens. There you go. Recovery. That's how the patient presented herself immediately after the stem cell thing. 9 months later, when we actually put in a regular cornea, and you find that what you have made actually is the corneal sheet. And here's the girl, 5 years follow-up. You can restore vision. It's easy because the eye is just as simple, the front of the eye is just as simple as the skin. When you can do skin biopsy and you can do skin transplantation, there's no reason why you cannot do corneal transplantation along these lines. That's by and large the story. Has she recovered vision? We have done this today on close to 700 people. The best corrected visual acuity remarkably improves. Does it survive over a period of time? When you do the surgery, let's say in 2005, what's the situation in 2010? And you do a follow-through, it seems at least 70% of the transplants must try very well. Do we have to only look for other organs and so on? Can't we do it from anywhere? It seems possible you can do it from anywhere now. That's what this fellow did. He took a cell from the skin and he said, what's the difference in terms of biology between the genes that are contained in there and the genes that are in the embryonic stem cell? Can I find out the difference between the activation there and so on, and put the necessary genes into the cell? And will they then become stem cells? And the answer to that is yes, you can. Shinju Yamanaka got the Lasker Prize, which is the equivalent of the Nobel Prize in Medicine last year, along with John Gurdon, who did the first experiment on tadpole. And our colleague Indumati has actually been able to do that on mouse, and she is now doing it on a patient, Delhi Prasad. Can we now make a skin cell, make it a stem cell, and try and see if we can make any corrections and so on? When you call me next, I'll talk a little bit more about whether we are successful or not. Thank you.